Good evening, everyone, and I hope you've been well. I hope you've had a very good start to the term. My name is Krzysztof, and today it is my pleasure and privilege to welcome Professor Sir Leszek Borysiewicz as our speaker. Professor Borysiewicz is the current chairman of Cancer Research UK and has also been the chief executive of the Medical Research Council, as well as a vice-chancellor of the University of Cambridge. We'd also like to thank all of our sponsors for making our events possible and for helping to finance the development of BioSoc. In particular, we'd like to thank BitBio, a synthetic biology enterprise, as well as CRISPR Biotech Engineering, an early stage genome editing company that also focuses on providing educational resources. Today, we are going to welcome a wonderful talk from Sir Leszek on the future ahead of us in cancer research, especially after the current situation, after the current pandemic. So without further ado, I'll now hand over to Sir Leszek.
Krzysztof, thank you very much indeed. Now, I have to congratulate you on perfect pronunciation, and even I can wrap my lips around Krzysztof, as that is my middle name as well. So thank you very much indeed for the kind introduction. So here we are at the start of 2021. 2020, only one word dominated the whole agenda: COVID. And yet the myriad of other diseases that we actually face and problems we have in relationship to pathology have not gone away at all. And in reality, what we are now having to look at are the consequences of the COVID pandemic and what that is going to do to set back both progress in helping to achieve major advances in cancer survival rates for the future. So turning to my first slide, what I'm going to do is to run through some of the issues that we face with cancer, some of the problems that COVID has caused, how we're going to respond to them, and why that response matters. Now, Cancer Research UK set itself a particular target. We've seen an improvement in cancer survival from where we look at survival for living with a cancer for more than 10 years. And going back to the 1970s, that was about 25% of all patients with cancer, excluding cutaneous cancer. The survival rate has now reached about 50%. That's good news. That's real progress. But we were anticipating that we were going to hit survival targets of 75% living 10 years or more with cancer by about 2034. Now, the scale of the problem is actually on this slide. This is the most recent data that's available because this data has not been skewed by the COVID pandemic, which I'll show you has the potential to skew these numbers quite significantly. But in essence, we're seeing around 370,000 cases of new cases of cancer every year. Deaths from cancer are about 166,000 per annum. And we estimate that 38% of cancer cases in the United Kingdom are preventable. It is the leading cause of death in the UK, above heart disease and dementia. But I don't like this sort of body counting, because if you take cardiovascular and heart disease and elsewhere, it's almost neck and neck. So the bottom line is it accounts for about 30% of all deaths in the United Kingdom. But the major piece of data that you have to look at is the IARC projection. That's the Institute for International Agency for Research into Cancer, which is a global epidemiological study group for cancer, shows that our current rate of cancer incidence is going to rise to 576,000 by 2035. That's a nearly 30% increase in the number of cancers we're going to see. And what's more, we can say that of that 29% increase, only about 1% is potentially avoidable. And the reason for that is that the aging population will mean we'll be seeing more cancer. This Thursday is going to be World Cancer Day, and therefore this global projection gives you the measure of what increased longevity in the whole of the world population is likely to see. That's a doubling in the number of new cancer cases by the year 2035. And this is going to pose a major problem in developing countries where the ability to care for cancer, if we're to hit such targets on a global scale of improving survival, make it extremely difficult. And World Cancer Day is there to highlight this particular issue. Now, the good news is more people will be surviving cancer. This is data taken from, based on a study by Bluthman in 2016. I pinched this figure from Shapiro's paper in the New England Journal of Medicine. And these are the projections of the number of people who will be surviving cancer. But here is the important area, that by 2040, most of those survivors are going to be in the 65-year plus age group. So not only are we going to see an increased prevalence in those living with cancer or having survived cancer, survival here being defined more or less by 10 years or more of living with cancer, that extended period of time is going to have a huge change in the way in which we have to deliver healthcare, both to cancer and the long-term complications of cancer going forward. So you're going to live longer, you're going to live with cancer, but that will also raise the issue that we will be seeing more comorbidity. Patients with cancer will be having other conditions, and they themselves will begin to impact on cancer. For example, any inflammatory condition, as I will show you later on, may well have a significant impact. Now, this improvement in the outcome of cancer treatment, you can date back from 1971 to 2010, 2011. And here you can see the general improvement that we're observing in virtually all cancers. However, there are the standard 5 cancers where we've seen virtually no advance in at all over this period: stomach, brain, esophagus, lung, and pancreas. And you can begin to see by the diameter of these circles that these are still common diseases, and a huge amount of effort has got to go into trying to improve outcomes in those tumors. So a lot of hard work needs to be done. For some of them, you will see, like prostate, we don't know, is this a real improvement? Is it that we're screening more people with relatively benign disease? Same questions have been raised around breast tumor, but the improvement, particularly in cancer of the uterus, the non-Hodgkin's lymphoma, cervix, larynx, and particularly bowel cancer, these are very real and very important for patients with the disorders. But problems remain. I could show you a lot of slides on socioeconomic factors and the fact they play in survival. But here's an odd one, this data taken from the United States, in that Black patients are still faring less well by 2016 than the improvement in mortality that you've seen in white, including Hispanic women. And why is that? There's a failure here to deliver appropriate healthcare. There's no other reason why these lines should be divergent. So we have a lot of problems to still overcome. And before we pat ourselves too much on the back, let's look at this data. This shows the variation in the United Kingdom in 2001 for the percentage of people who would survive 1 year of a cancer diagnosis. Huge variation, ranging from 50% to 68%. Now, the good news is by 2016, these bands had narrowed. That means that the development of multidisciplinary teams means that wherever you are in the country, you are likely to be within a very tight band now, being able to receive appropriate cancer treatment. And this is visually seen in this Office of National Statistics map. And if you're interested, you can go to their website and see these from 2001 onwards, that around the country, there's almost uniformity in how 1 year survival is going. But 5-year survival and 10-year survival still shows unwarranted variation in outcome, even in a country like the United Kingdom. Now, one of the studies that we support in Cancer Research UK is what's called the Survey Mark study, which was reported in the Lancet Oncology in 2019. Here we work with countries that are very similar to the United Kingdom: Australia, Canada, the Scandinavian countries, New Zealand, and Ireland. These are countries that collect data in almost the same way, so allows for real comparisons to be made. Now, all I'm going to say to you is the arrow that it represents the United Kingdom in the 5-year net survival rate, and this shows an improvement from 1995 to in 2014 in all these countries, but which arrow is found at the bottom in terms of 5-year survival rates. These rates, for instance, for rectum, bowel cancer compared to Australia, stomach cancer, esophagus, lung, whichever you choose, an ovary, you will see a big difference between these countries. So don't pat ourselves on a success story too much, because we're not catching up these countries. We're applying probably the same methodology to gain improvement, but we're not gaining the improvement that we would hope to see, because we eventually want to reach a point where all of these countries are operating at exactly the same levels of survival. So huge amounts of work still to do. And all that was great. And then along came COVID, progress being made, but how has COVID impacted the cancer pathway? Well, firstly, does COVID pose a risk to patients with cancer? Now, this is a survey that was published in Nature by Williamson and is based on the UK primary care records of 17.3 million people. That is 40% of all NHS registered patients. And what they began to look at was what was the risk if somebody contracted COVID, on the hazards ratio on COVID mortality based on underlying conditions. And as you can see on the left-hand side, I'm not expecting you to read all of this in detail. You can begin to see that a myriad of conditions have an impact. So you will see organ transplantation here at the bottom has a significant impact, and you'll see diabetes, in particular uncontrolled diabetes, has a very significant impact. Chronic cardiac disease, but hypertension and high blood pressure itself does not have that impact. When, however, we begin to focus in on the cancers, what we see very clearly is that the hematological malignancies carry a great risk, as well as the early diagnosed cancers, rather than those who have already that have received a cancer treatment in terms of the hazard ratios for those conditions. This is about a 2-fold hazard ratio. And when we look at data that's been reported in trials and other published series, we tend to see that the case fatality rate for patients with cancer receiving, then being infected with COVID is around 23%. That's higher than is observed in other conditions. And again, you can see what comes through are the hematological malignancies, but particularly thoracic malignancies and brain tumors are coming through, almost certainly by the aggressive nature of treatment that they receive. So COVID directly impacts on the capacity of patients to survive, and it certainly is a detrimental combination. But what about the impact it's had on healthcare services? This is the data on the impact on cancer screening. The bottom line statement is in the United Kingdom, 3 million fewer people were screened between March and September of last year. That's not even with the second wave being taken into considerations. And the number of people starting cancer treatment following screening in England was down by 58%, and in Scotland down by 71%. Now, I'll just leave you to ponder the impact that that's going to have in years to come, when the numbers aren't going to be large enough to be seen on the sorts of graphs that we're shown on television night in, night out. But these are going to have long-term consequences. And as we go on, we will see that there is an impact on urgent cancer referrals. And this impact was particularly visible in March to April. So we're anticipating that similarly this data, and we already have some preliminary evidence that that data is already dipping with the second issue. Now, the good news is we caught up pretty quickly to reach the same rates of treatment as before, but the catch up on the dip, the area under this curve, has not been caught up. And so 400,000 fewer people had received urgent appointments for care. And by different cancers, you can just read that for yourself as to what's seen in England and Wales. We don't have equivalent figures for Scotland and Northern Ireland. But again, on this graph, you can begin to see that the referral types related to the most common cancers have gone significantly down. And I particularly point this light blue line on the lower GI tumours and breast tumours that we're seeing, as well as the dip in urological cancers. And diagnostic tests, this is what really plummeted. 4 million fewer key diagnostic tests were conducted between March and November of last year. And while radiology has suffered in particular because of the need to clean equipment, and it's had to slow down the use of radiological equipment, endoscopy in particular has gone right down. This is because this technology is at particular risk of spread of COVID. So the number of patients beginning treatment of any sort is now down by nearly 17%. And you can see that more or less equivalent across all parts of the United Kingdom. And we can go through this by site, and we can look at other times. The time people have to wait for a diagnostic test is also considerably down. And in particular, waiting for endoscopy, a 6-week wait for an endoscopic examination has increased 8-fold. So we're seeing earlier pauses to the screening programs, with fewer patients beginning treatment for cancer following a referral. Urgent suspected cancer referrals were knocked back. Key diagnostic tests were around 8 to 11 times more patients on waiting lists. That's 700 to 1,000% more. I've been waiting for 6 weeks or more, and the monthly numbers of patients beginning treatment for cancer are huge. We believe that even in this first phase of the disease, an accumulated 40.9 thousand fewer patients are in this backlog. So this is going to have a big impact. And therefore, conducting research, if we're to hit this target of 3 and 4, that target will have been set back. There's no doubt about that. But what we've got to be able to do is to ensure that the research that is undertaken in cancer biology and in understanding the optimum forms of cancer treatment is going to be absolutely essential if we're to get back on track with the target that we've set ourselves, and one which I think most people would very much like to see us hit. So where are we with current directions on cancer research, and where are the directions likely to go in the foreseeable future? And here I'm going to focus a lot on basic research and asking some of the fundamental questions which can result in improvements in treatment, maybe in 4, 5, 6 years' time. Now, to get us onto the same page, let's just understand that while for NHS and for clinical care purposes, we often consider cancer in terms of its anatomical location. So breast cancer and colon cancer not considered. But we can begin to see a convergence of ideas in the Vogelstein model that cancer is a somatic genomic disease. And Vogelstein raised his hypothesis in that from normal colonic epithelium, you can begin to move towards an adenoma because of certain mutations that actually occur in certain gene pathways. as those changes occur and cells replicate, the incidence of somatic mutation increases and other driver mutations are added. And you can end up with larger adenomas, still benign conditions, but now you have really disorganized cell growth. But with further changes, what you now can get is the invasive disease. And this accumulation of mutations is something that we're seeing time and again with cancers. And to study this, the Tumor Genome Atlas was created, where 10,000 tumor samples from 33 cancer types were pulled together with using 26 different software tools. And what we began to see is that of all the genes on the whole genome that is really important, in all cancers, the mutations that really began to matter were down to nearly 300 genes, the so-called driver mutations. And those driver mutations are obviously ones that we need to be able to action. And here the good news is that when we look at all of the medicines in cancer trials, I don't expect you to read the details of this, nearly 57% of the common mutations that we see in driver genes are actually covered by drugs that are already in development for those tumors. So we're moving to a situation where precision medicine is likely to take root in cancer care, because we're going to be able to equate genomic change much more to the type of treatment the patient is going to receive, and maybe even more so than just the anatomical location of that tumor, even though that matters for individual patient care. So the precision medicine may be more closely targeted to the genomic changes that have taken place if one begins to assume that cancer is a somatic genomic disease in its underlying etiology. But to give you some idea of the issues that arise, I'm going to go back to a series of studies that were conducted in Utrecht to just begin to look at a single adenomatous polyp, and taking 4 cells from that very same polyp as single cells to grow them up through a few divisions into organoid cultures, and then to begin to look by mutation analysis as to what are the mutants that have arisen from a single polyp. How much variation is there in those early stages of disease? And the answer is an enormous variation. You can see this just in the number of mutations that are actually seen. Derived from position 1, you can see there were 75,000 mutations in the cells that were there, 30,000 from deletions, a few insertions, all sorts of variation, compared with the same tumor, with the same adenoma and cells derived from that adenoma, much fewer deletions and inversions occurring in these other sites. So the huge variation, even from a single site, is absolutely enormous. And when we start looking at epigenomic markers, there are even more changes. And these changes take no account of stroma, because they've actually been separated cells that have actually been grown. And this paper by Roering, to me, really exemplifies the scale of the problem that we're dealing with. And added to this is that when you begin to look at different cell lines derived from different patients, even though they've come presumably from a single set of mutations arising at the time the polyp arose, we can already see that resistance to each of the common drugs commonly used varies from tumor to tumor, in patient to patient, and from cell line to cell line. This is important because what this is showing us is that there is no combination of cancer chemotherapy that could have been used to treat that patient to make sure that every single cancer cell is killed. It would have actually killed the patient in the process. And it also suggests that many of the resistance markers to conventional treatments are already inherent and likely to be inherent by the time the disease is diagnosed in an individual patient. So we've got some serious problems ahead of us with real implications of basic biology when we start transferring this to management of a single patient. So the earlier we can go back, the better. And what could be better than looking at preventable causes? If we begin to look at the number of cases per year that by attribution of causation to different factors, we can see the number of lives that could be saved. So if tobacco was banned tomorrow and there was no tobacco available, we could see that maybe 55,000 cases of cancer per year would actually be reduced. In a variety of organs, it isn't all lung, it's actually the impact tobacco has on cancers at other sites. But coming up increasingly and close behind is overweight and obesity. Now we know and have a good idea how smoking is related to the development of cancers. It's a set of complex carcinogens, and we know that we can take public health measures to reduce smoking. But smoking rates do not come down on their own. Merely telling people that there is a risk, it does not work. Nearly every major reduction in smoking rates on this graph has actually happened because of government interventions, either by raising taxes or by making it socially unacceptable for people to smoke in public places, marking cigarette packaging. The good news is that this downward trend continues. And interestingly, one of the positive effects of COVID is we're seeing more and more people wanting to give up smoking, more of anxiety around the COVID situation than maybe even thinking about cancer. But we'll take anything. And this has to be a remarkable success story when we go back to the 1970s and look at the rates of smoking that existed then. Now, if we project this further forward, it does actually mean that with a rising epidemic of obesity, obesity will overtake smoking sometime after 2030-31 as the commonest association with cancer. But this association is quite complex because obesity does not affect every cancer. You do not see obese patients with lung cancer. Only 13 different cancers actually show an association with obesity. Bowel has a particularly strong association, and breast is complex because there are differences for premenopausal and postmenopausal breast cancer. So a big question has to be, why? How does obesity cause or is causally associated in any way with cancer? This question is one that is really quite a challenge to be able to take forward. And I would be probably quite okay to say that Cancer Research UK is going to focus a lot of effort and energy in trying to understand this conundrum. Now, the epidemiology is absolutely clear-cut. There is increased cancer incidence in obesity, and there is some evidence that even when you've got cancer, there is more aggressive progression in cancer in obese subjects. But why? And there's a very good set of papers that are coming out increasingly towards the latter end of 2020 that are giving us some hints as to what could be the molecular mechanisms that could underlie this particular susceptibility. It is very clear that it's going to be a multifactorial issue. It's going to vary from cancer to cancer, and it's probably eventually going to be linked to the type of mutations that we actually see in cancer cells. There is increasing evidence that systemic tissue and cellular metabolism of glucose, and maybe even more particularly of lipids, may be playing a key part in this. In other words, it's not just the shared genome of risk, It is something about the obesogenic environment that is present in these patients that is actually causing or is linked to the more aggressive type of tumor that is observed. And one thing that has been shown epidemiologically is that adiposity-associated insulin resistance or reduced glucose utilization is linking to a poor outcome. And that's a good hint that, again, we're dealing something with the environment in which the tumor finds itself. But we're also seeing that adiposity-associated changes, not changes, in immune function, especially for CD8 cells, T cells, may play a very important part linked together with the adiposity-associated insulin resistance. And here, the tumor is not just a passive, but could be quite an active engager in blocking that response to the T cells. And altered tumor metabolism towards glucose and lipids may be playing a very active part. Now, I'm going to refer to a series of studies published right at the end of December, just after Christmas of last year, about a month ago. In these experiments, what Ringel and colleagues at Harvard did was to take wild-type C57 black mice and to feed them on a control diet or a high-fat diet for 8 to 10 weeks. And then they injected subcutaneously tumor cells of different sorts: MC38 breast cancer cells, melanoma cells, or lung cancer cells. And as you can see, not with every cell type, Lewis lung cancer, carcinoma cells show no distinct difference, but MC38 or melanoma or breast cancer showed that in the obese mice, you get what you'd expect, that the tumor size increases, the tumor volume increases more rapidly than those mice fed the control diet. But if we look at where MC38 cells were injected subcutaneously, if we knock out the T cell receptor gene, we can see that the tumor progresses at exactly the same rate as it does in the mice fed a high-fat diet. So whatever this phenomenon is, it's actually a T cell mediated phenomenon in these mice. And furthermore, compared with isotype controls, if we now deplete the T cell population with anti-CD8 antibodies, we can see that it's the CD8 cell component that appears to be playing a major part in restricting the growth of these tumors. They then were able to show that in the tumors as they were growing, the high-fat diet reduced the number of tumor-infiltrating lymphocytes, whilst numbers of infiltrating CD8 positive cells were normal in other tissues. So something is going on in the local environment between the tumor, the stroma, and the capacity of T cells to actually come into that tumor that is important. Now, there are a large number of component parts, and I'm only picking up one that matters. What they then began to look at was that oxidation of long-chain fatty acids appeared to be particularly important in this activity. And it has been shown through the Tumor Genome Atlas that a particular enzyme, PHD3, which regulates mitochondrial import and oxidation of long-chain fatty acids, had a particular impact. So what they did was to take the PHD3 tumor in the tumors that were being used, the MC tumors in these experiments, and began to overexpress PHD53. And when they expressed this, what they actually showed was that in these tumors, the average number of CD8 cells actually gradually increased, both in the control group, but particularly in the high-fat diet fed group, over that found in tumor in the opposite flank, which just had empty vector. Again, this phenomenon was was abrogated by depleting CD8 cells or by taking out all T cells. But they then did something else. They noted that an association was found in the tumor genome atlas patients with human colon cancer. And what they then went to look at was to see that if PHD3 expression, which was seen in normalized counts, linked to the body mass index of these patients. And those patients with a body mass index of greater than 30 had far lower rates of PHD3 expression, and also that the number of CD8 cells that were present in the tumor as opposed to those who had a body mass index less than 35. And particularly in those tumors that were seen to be relatively cold with regard to the number of CD8 cells that were present, you began to see far greater expression of PHD3. So this has led to a potential hypothesis that looks quite plausible. If PHD3 is increased in the tumor cell, free fatty acid transporters are less active in that tumor cell. That increases the ability of free fatty acids to be present, can be taken up by T cells, and you increase T cell activity and number locally. If you have relatively low expression of PHD3, as happens in some of these tumors, you get more of the free fatty acids entering the tumor cells, outcompeting the T cells, making the T cells relatively energetic. Now, that's just one experiment in a mouse model of disease. But is there something else that begins to point to this, to the potential importance of high fat? And this was a study conducted again in January of last year by Zhang and was reported in Cell Metabolism. And it looked at the impact of obesity on the leptin STAT3 fatty acid oxidation pathway. Basically, what they showed was that the presence if we remove STAT3 from those situations, we actually find that the number of CD8 cells also increases. And the common pathway is again built around fatty acid oxidation. So in many ways, even here, the leptin receptors present on CD8 cells may well be having a major part to play, and again suggesting mechanisms whereby the disordered lipid metabolism in these animals may be causing these problems. Now, most of these studies are still based in animals. We all know how notorious animal models of malignancy are, but they're beginning to start shining a light that underneath it all, there may be mechanisms which we could use to enhance and to improve from what we learn about the problem of obesity in relation to cancer, that what we learn could actually lead on to new treatments. And this is really quite exciting work and an exciting area of development, and may well tell us also an awful lot more about tumor T cell biology as to what might be necessary to maintain an effective immune response, which may play a part in immune surveillance, even from those patients who may be recovering from first treatments of cancer. So one of the questions which we often face is, what are going to be the really big questions facing cancer? So before we went into lockdown last year, we undertook an engagement with the National Cancer Institute to set up Cancer Grand Challenges. Now, we've had several of these for 2 rounds, and already 7 major challenges are already underway. But with the National Cancer Institute coming in with $250 million and our commitment to actually engage with this process, we undertook to have town hall meetings around the world with cancer experts, asking the question, what are the most important research questions that we need to be able to pull together? And then we appointed a body under the chairmanship of Paul Nurse to begin to pull the learning from the variety of meetings that we had held, to synthesize this down to a number of challenges which we can set the research community. And with NIH, we're committing to funding 3 projects, again, this time for the 3rd round of these challenges, where we will fund them to the tune of 20 million sterling for 5 years, or $25 million. We're an international team, pulling scientists from many different countries, can come together to address these major challenges. And this is the upshot of the work that they've pulled together. Understand how cells and tissues maintain normal phenotypes whilst they harbor oncogenic mutations. And then how do they transition to become a tumor? Can we deliver macromolecules to intracellular targets? Because we know many of the targets in cancers are going to be intracellular, but a big problem is the delivery of macromolecules to the intracellular environment. Cell senescence. How can we exploit that to improve cancer treatment? An important one, which you may want to come back to me, are the risks of e-cigarette use. So quite a different challenge. Inflammation causing cancer, how does it do it? We've known through the history of cancer that inflammation is clearly associated with cancer, but by what mechanism? And how can we block it? Solid tumors in children, particularly developmental tumors, are a different type of tumor that needs targeting differently. Extrachromosomal DNA and its role in cancer. Cachexia. We all know that losing weight is a common symptom of all cancers. And yet, to this day, we do not understand why patients get severe cachexia or how we can reverse it, as this is very often a problem as cancer progresses in so many patients. And then maybe most intriguingly, to identify and target dormant cancer cells. I'm going to just pick 1 or 2 of these challenges because I want to highlight how they will all interrelate together and how the basic molecular mechanisms that exist are likely to shed light on several of these particular perspectives. How cells maintain normal phenotypes. This is work coming from Fitzgerald and colleagues here in Cambridge. And they began to look in this picture at a square millimeter of normal esophagus. This is not malignant esophagus, but they started to look at driver mutations that are common in esophageal cancer. The question is spread over a millimeter of the surface area of an epithelium, how many actually these mutations do you see? Even in tissue taken from a normal 20 to 23-year-old male non-smoker, you can begin to see that here you've got a P53 set of mutations in an island in this epithelium, and you can see that there are individual cells already harboring that area. Similarly, you see this in a female non-smoker as well. But when you're starting to reach my age around here, you begin to start really wondering how on earth not everybody actually has esophageal cancer. So many of those cells are already carrying driver mutations, and yet esophageal cancer, thankfully, is a relatively rare disease. So how does that progress from these changes into something that is going to have the cancer phenotype? Or maybe more importantly, what mechanisms exist to repress those changes in such a way that the individuals don't develop esophageal cancer? And this phenomenon is seen in almost any tissue you wish to study. I've just picked this study as it's so graphically evident from this slide. A very common phenomenon, but one we don't understand. Are these cells eliminated? Are they coped with? Are they contained? What is actually going on? But one of the big issues that we face is dormant cancer cells. if we take a patient with cancer and we treat them, we see basically progressive disease, or the treatment can convert that disease to stable disease, or to get a partial response, or to get complete response. The 4 outcomes that we would broadly like to see. And in these, we can begin to put them on a sort of linear scale as to the residual tumor volume that remains in patients in these areas. So some will still have a slightly increased tumor volume, normal. Others, it will show a major decline. Now, in all of these patients, even those potentially with a complete response, there are residual cancer cells, the so-called persistent cancer cells that exist. And over time, these can result in a relapse. But are they truly dormant cells, or in other words, that they are so quiescent that they can remain completely locked in for a long period of time? Or what is the state of the biology of these cells, and how are they able to sustain themselves over prolonged periods in the host, both under control in some instances, and then that control is lost in the individual such that the disease recurs? And the question here is, are these cells, do we have dormant cells actually in the original site of the tumor, which then spread? And in other words, that dormancy is carried because they already have a characteristic of dormancy. Or do we induce that in those individuals who get to a complete response, where not only might there be the odd persistent cell, but some of those might actually move towards dormancy? Is this process passive or is it active? Now, the theories on cancer dormancy have come and gone over probably for as long as cancer has been studied. And first of all, it was thought to be a relatively passive process. Some of the theories were around angiogenesis, in other words, that these tumor cells in where they find themselves located cannot actually get a blood supply, therefore they can't grow, and therefore they are halted from growth. Or that there is an equilibrium state, that there is immune recognition and surveillance, and these cells are eliminated, and if they grow and divide at even a slow rate, there is a sort of equilibrium that's reached. And obviously, if you alter that equilibrium in any way, by a new mutation in a cancer or by treating the patient with an immunosuppressive, you can suddenly get the tumor growing out. Or the microenvironment, where a tumor metastasis may find itself, or where these dormant cells that are spread probably very early in the disease process might actually cause a growth arrest in these cells and keep them under control until that microenvironment changes. All of these are passive as far as the tumor is concerned. But what we're now learning is the tumor is not a passive passenger in this process. It actually participates in it. And the very definition of dormancy is very difficult now to undertake. If we have a look at particular markers of dormancy as found in this paper by Rissen, who summarized an enormous amount of very good work in this area, you can have fully dormant cells. These can survive, they can self-renew, they have more or less blocked proliferation, and they don't differentiate. Or they can be quiescent. There is some proliferation, there is some differentiation. That survival and expression of different markers may well mark them differently. And the outcome of this could be that these cells could differentiate, or they could go into senescence, which might favor their clearance, particularly by the non-adaptive immune system. But in all of them, cell cycle arrest appears to be a critical component. So what we're learning is that cancer cells may well respond to some of the developmental cues we see in embryos, and particularly developmental cues as we see in restraining adult stem cells in the normal scenario, both hematopoietic and other tissue stem cells, because we don't want these stem cells overgrowing. We also now begin to understand, particularly from hematological studies, that active regulation of cell signaling enables survival of cells even after treatment. So this survival after treatment, again, is not a passive activity, but is due to changes in that tumor cell. Avoidance of immune clearance may well be important. We find an awful lot of these have downregulated signals, such as downregulated MHC class 1. They have downregulated receptors on their surface. And the tumor, far from being a passive passenger with the microenvironment, actually has the capacity to change the stroma in which it's found. And so dormant cells may survive for long periods by actively co-opting growth arrest and survival mechanisms that might be used in normal development. And in the same way as that normal development requires the stroma, the tumor microenvironment, not just the tumor cell itself, may play a very important part in the continued survival of dormant cancer cells to come out much later on. Now, this slide summarizes comparatively dormant hematological stem cells and what the characteristics found on dormant hematological stem cells, which again have got to be very tightly regulated, as opposed to dormant cancer cells. And what are the pro-dormancy niches that we actually observe, and the cell types that are really important in maintaining their dormancy? And here we can see mesenchymal stem cells will play a part, the sympathetic nervous system. Osteoblasts in particular are important. Endothelial cells and Schwann cells, particularly in the pericytes that have been found in relationship to secondaries in murine tumors. Interestingly, osteoclast recruitment may well be a trigger to reactivation. And sometimes pro-inflammatory loops may be important. Here, a very interesting set of experiments to release cells from dormancy have been found through sham surgery in the mouse, that if you induce inflammation, where there are an awful lot of neutrophils around, you end up with the development of DNA traps. And these trigger the awakening of dormant cells through the release of neutrophil elastase and MMP9 enzymes, which promote laminin and alpha-3 beta-1 integrin dependent activation of the signaling axis. And it also involves kinases such as ERK and FAC in this reactivation process. Now, I don't expect you to take away all of the detail on this slide. And if I say this slide is a gross oversimplification of where the situation resides, but I want you to take away the sheer complexity of what we have to overcome and to understand in order to be able to target treatments appropriately to be able to either eradicate or to maintain cancer cells in a dormant environment to allow people to live and survive their cancer for longer. And if we're going to identify these dormant cancer cells, we need to be able to identify what are the mechanisms that may be important in maintaining that persistent or dormant state. And these come down to four particular mechanisms. Slowing down proliferation, and here you've got adaptive mutability, you've got alteration of the microenvironment and elsewhere. You can adapt cell metabolism, so mitochondrial respiration, the dependency on ribosomes, the oxidative trade-offs that you begin to see. You can actually change the cell identity. Here is a very important phenomenon that is not very well studied, is the epithelial mesenchymal transition that is seen in certain tumors. Or the cancer cell can hijack the microenvironment to enter into a kind of symbiosis to maintain the persistence of these cells. And immune suppression is a key component part, because otherwise these cells could be eliminated by immune surveillance mechanisms. Now, we don't know which of these are important. We've got hints from animal models and from human studies that different ones work. For instance, there's been the age-old anecdotal study that you probably also have heard, that if you take a patient with a relatively late-stage disease and you open up, suddenly the tumor progresses. Well, there are one or two mouse studies which suggest sham surgery can enhance progression. Is there something in that old anecdotal tale that needs further exploration? I don't know. But what I do know is an understanding of this particular phenomenon will tell us more about tumor cell senescence. It will tell us also how we regulate those cells which may carry driver mutations, but not progress to malignancy. And therefore exploring these mechanisms at the level of the tumor cell, at the level of the persistent cancer cell, and its interface with the microenvironment and the macroenvironment of the metabolism in the individual patient or animal may be absolutely critical. So there are the challenges. Any one of you fancy creating a group that might actually get a very substantial award or participating in these studies? The website is there, and the Cancer Grand Challenges, which you can get to. Have a look at it. See if you can engage in these areas. Because by working in larger groups, we're going to be able to tackle several components of this at the same time, which individual project grants just cannot begin to address all of these components in relationship to these diseases. So they're complex questions and there will be complex answers, but that complexity can be overcome by increased collaboration. So my conclusions are that overall, we have to pat ourselves a little on the back. The considerable progress has been made to enhance outcomes for patients with cancer. It's good, it's not good enough, and it's got to be better, but it is improving. COVID, yeah, as I was asked to comment on that, has impacted adversely on progress towards better outcomes, not because of treatment failures, but actually because of a whole systems problem, that the system has been overwhelmed by COVID, and therefore delays are the real issue. Catching up on those delays may seem a simple thing theoretically, a very difficult one to achieve in practice. But if we're to catch up to again reach those targets of 3 and 4 surviving, I think we've got to really grasp the challenges to understanding mechanisms of carcinogenesis and particularly disease progression and what can actually prevent the longer term recurrence of disease in patients who have successful treatment. And I think there are key areas to address. How are risk factors? And I'm going to pick out obesity because this is a real conundrum, can be associated with cancer induction and progression. Because if we can identify those and we can identify the mechanisms, not only might we be able to help the obese patient, but we may be able to learn what factors are important, maybe in that CD8 tumor interface that we could adapt and utilize to enhance the importance of that CD8 surveillance in the condition. Disease progression. The primary, unless it's in a critical site, may not kill you. It's the site where most of the mutations will take place. But very often it's metastatic disease or disease recurrence or long-term control of the disease really matter. And here, understanding the nature of cancer cell dormancy, persistence, and what are the controlling mechanisms of that are going to give us those longer term survivors and increasing those so that we can indeed hit our target of 3 and 4 living for 10 years or more. So if we are to achieve an improved 10-year outcome, then addressing these basic questions and translating them into clinical action will be essential. And it will need all of us, I hope at least 1 or 2 of you are listening to this today, to be inspired to recognize that we've made huge progress and we're on the cusp of making even more progress, which will be of direct benefit to so many others, not just in the UK or in developed countries, but increasingly important in developing countries as they're going to face a pandemic of cancer in the years to come. Thanks very much indeed.
Thank you so much, Sir Leszek, for that, well, at the risk of sounding very pompous, for what was essentially a masterclass intertwining aspects of cell biology and basic science, as well as wider issues regarding translational medicine and applying that to clinical and medical practice. So I'd like to open the floor up to questions. Feel free to ask questions in the YouTube live chat. I'll be monitoring those. And we do actually have our first question from the Zoom chat. Shrez is asking, could there be a connection between the microbiome and cancer, perhaps contributing to the relationship between obesity and cancer?
The answer is yes. One of the grand challenges that was funded in the last round is to better understand the microbiome in relationship to cancer. So what we've had so far is we know that the microbiome can affect the outcomes of cancer positively. Changes in the microbiome can actually be associated with an increased incidence of certain types of bowel cancers. That's a pretty local effect. But intriguingly, the nature of the microbiome also defines the response of certain tumors to the drugs that the patient will receive. So the microbiome is playing a role. What we don't understand is what the role it's playing. What also we don't know is in that obese environment that you find in that C57 black mouse, or you would find in a patient, in an obese patient, it could be that in reality, it's the microbiome that we've been playing a big part, as we know in diabetes and elsewhere, in defining the metabolic consequences of that disease. So we may be studying the effects that we're seeing on the cancer cell, but the causation of those effects might actually reside in the microbiome. So yes, we need an awful lot more work. We're funding an awful lot already on the microbiome, but if anyone's interested in pushing that idea further forward, believe me, we are very receptive to it. There's an awful lot that can be learned there, and more importantly, could we manipulate it to an appropriate scenario whereby we can begin to get real patient benefit?
I think that's a very relevant topic that many people are interested in. For example, in our BioSoc committee, there are 2 people who are definitely going to go, as far as I'm aware, into microbiome research.
Yeah.
So, perhaps following up from that question, do you think we'd have to be aware of whether that research with the mice, whether they controlled for the composition of the microbiome or not?
There was no material presented in terms of the metabolomics to look of those mice. So we know the answer. I can't see it in the publication. I haven't written to them about that, but it's an intriguing study, and it's one that I'm sure they will have the material to be able to study, because they would have taken the relevant serum samples, but it needs some metabolomic work. There has been some metabolomic work done in relationship to obesity and cancer. And again, what's mostly been noted is there are changes, but people haven't yet associated any of those changes very positively with the microbiome, with particular aspects of the microbiome. But at the other end, we know the microbiome actually affects so many of the metabolic pathways that we engage in. It's difficult to almost say there will be no association. There will be an association. The question is going to be how significant it is. And then, being a practically minded individual, I want to know, can we manipulate it?
So looking into its translational significance. We have our first question from the YouTube live chat. Thank you for the excellent talk. Do you think an understanding of how cancer evolves in response to treatment will be incorporated into how the disease is treated?
The answer is absolutely. We're going to see, I'm now trying to crystal ball gaze, so this is a personal view, and you have to take away a lot of these selected areas are personal. There's so much going on that it would be impossible to cover the whole area. What we're seeing is less and less reliance in basic research on mouse models and more on actual patient material. We can do so much more now with the real material that we face, particularly in wild-type tumors, than we were ever able to during the time when I was starting research in some of these areas. The importance here is that we have, therefore, the capacity that everything we do can be related back to patients. And if we really believe that ultimately cancer will lead the charge for personalized medicine, and you can see all those drugs that are in trial, we have an armamentarium of drugs and potential interactions. The personalization of understanding what the driver mutations are to modalities of treatment that can be adopted in individual care is going to require an enormous amount of AI work, of machine learning and elsewhere, because the complexity of it now is such that it's going to be very difficult to thrive. But if I threw a challenge to you, that in a future that I might see it in 10 years, 15 years time, we will have it, it'll be very difficult to just conduct trials as we are at the moment on breast cancer patients, because they will be molecularly defined more and more. And it won't just be estrogen receptor positive or triple negative cancer groups. They will be defined by 15 different mutations. And oddly enough, those mutations in a colon cancer in Hawaii may have more relevance to a breast cancer patient carrying the same mutations than the equivalents to a breast cancer with different mutations from the same country. And so we're going to see more complexity, and we're going to have to learn more and more from individual patients and take that individual patient learning and be able to group and synthesize it together. So I think data is going to become critically important now in piecing together the molecular learnings that we're going to get, the potential for which treatments could actually be applied and are likely to be most effective. But it's going to throw a big challenge into the clinical trials agenda. How do you actually conduct trials when there may only be 5 patients in the world with that particular combination of mutations? Now, we can overcome those challenges, but it's an exciting future where we can begin to do more and more for the individual patient. And that individualization, I think, is going to become the future and the future target for in most cases. I hope that addresses the issue that was being raised, because it is a very important question for the future of cancer care.
I think that was an excellent answer. Well, really, what I took from it personally is that what we're looking at is individual patients and individual tumours, rather than having a blanket approach, and making sure that our models are actually relevant towards the particular patient being treated.
Yeah, and also remember that where it's going to start getting complex is, to date we've just focused on the tumour. Increasingly, we're going to have to focus on the stroma of that tumour. Yeah. One of the grand challenges that's currently being conducted in Cambridge is to start drawing a 3D atlas of what does a tumour look like. You know, it'd be amazing that to this date we have these views on a slide or whatever, but what's the endothelium like? What are the mesenchymal cells that actually surround a tumour? How much has the tumour affected those? How much does the mesenchymal cell affect them differently? We know from hematopoietic, for studies in hematopoiesis, they have a huge impact one on the other. And this interplay is not just a passive interplay, it's a very active interplay. So I see we're going to get an awful lot more data coming through from studies both of the tumour cell and the environment it's in, but also here now we're going to start getting tissue-specific localization of, for instance, does it matter whether a tumour cell lands in the liver and lung to develop a metastasis, or whether it ends up in skeletal muscle, when we know it's very rare to have metastases, even from common cancers at particular sites. And skeletal muscle is a great example when you think of how much muscle tissue we all carry. So what is it about sites that might be particularly important in progression of disease?
I think, as far as I'm aware, that's a group at the Cambridge Institute that has received funding, particularly for this 3D project, right?
Yeah, that's right. And they're doing a great job. Greg Hanna is the team leader there. Mm-hmm. But again, those of you who might be interested, go and have a chat with them. They are really engaged in trying to understand this very basic mechanism of how we get there. And for an immunologist, which is where I started, one of the most intriguing things is, how do we actually get those tumor infiltrating lymphocytes there when you haven't got an intact endothelium, and maybe all those pathways by which lymphocytes egress out of the circulation may not apply in a tumor. The tumor circulation is a static sort of pool of blood. It isn't actually a pumping active capillary bed in the way that we understand. It's an endothelial capillary bed that's been built up by induction from the tumor itself, and very often fails because you get these huge necrotic parts of tumors where the blood supply has gone on. And the tumor cell adapts as you saw in that STAT3 study, that it very often adapts by, for instance, using the glycolytic pathway rather than actually using the aerobic metabolic pathways. All intriguing questions. All I wish is I was just 40 years younger and being able to really just enter this field. It never, to me, it has never been more exciting than it is now.
As much as I'd love to ask more follow-up questions on this topic, I should probably move along to more questions from our YouTube live chat. So we have one from Milena Malharek, who asks, given that adiposity-associated insulin resistance is linked to poorer outcome, has there been any research on whether drugs like metformin can be used as adjuvants or in prevention of cancer?
Yeah, there are studies going on with that, and there is also evidence from studies that were actually grouped together from an Institute of Medicine report into modification. For example, a simple one, does it actually help you to lose weight if you've actually been diagnosed with cancer? I mean, it's a relatively simple intervention. Am I putting backing? There is some evidence that that is probably a good thing to be able to do. So the answer is, I don't know the answer to that directly, but I do know that studies are in progress to begin to look at how we can begin to interfere with this interface.
And perhaps going back to the general idea of microbiome interactions, but in a different context, another person on the live stream asks, well, starting off with, thanks for a very informative talk. Intratumoral bacteria have been reported in many tumor types. What's your opinion on their roles in cancer progression? And do you think this field has a future?
I think the field does have a future because they do exist in these scenarios. So they've been well identified. These are not artifacts. It appears to be a very real association, but again, we don't know. So it's a little bit like the microbiome's association with intestinal epithelial cells that occurs in the normal environment. We don't understand fully how much of an interaction, what that interaction does. I cannot go back to the a priori assumption that if you've got these living organisms in direct contact and engaging, they're going to have an effect. At the crudest level, if nothing else, they're going to induce inflammation. Inflammation itself can actually induce changes in the tumor. It can actually be associated with progression, as has been shown by these nets that have been identified when neutrophils, as they die off, release elastases and cause problems. So yes, there are going to be associations with them. I really do believe that, and they're definitely worth exploring. But at the moment, it's still early days. People are still reporting very much the phenomenon of finding bacteria in tumors rather than how they might actually be going together. And certainly they're going to affect the metabolism of both.
Maybe a personal follow-up on that. A very naive question perhaps, but is it just bacteria that have been found within tumors, or is it other kinds of microbes as well?
Oh, you're bound to find other types of microbes. I'm sure if you started looking for viruses and things, you will find them, but it's mostly bacteria that have been particularly raised the issue, because up until now, I think people have probably realized that they were there, but they've kind of been ignored as to whether they play an active role. And I think what's changed is people are now, after all the work on the microbiome, starting to pay much more attention to organisms that are there. And secondly, we have the mechanisms now to measure them, because some of these will not culture in the conventional microbiological culture systems. You actually find them through molecular signatures rather than and metabolic signatures rather than necessarily find them elsewhere. So we now have the capacity to study them. And I do think some interesting and important work will come from that.
So it's a combination of both interest and the technology that's come in.
Interest in technology is always the same area. And the fact that people have been prepared to challenge a paradigm, whereas previously they would have just said, oh, it's an artifact. I mean, you know, you must have got, it was just a dirty slide or whatever when you were looking at it, and it was just ignored. But I think there's enough evidence to say that this could be a very real phenomenon. I'm certainly quite intrigued by it.
Milena sends her thanks along for for your answer to her question. And we also have another question that relates more to the current situation of COVID-19 from Paul Carter. Thanks for a great talk. What do you think underlies the strikingly poor COVID-19 outcomes in hematological malignancies? Is it likely that associated mutations cause a hyperinflammatory response?
That's an intriguing suggestion, actually. The convention is that it's in these patients that they have particularly disturbed immunity. So one of the areas has been that they've been unable to develop the natural immunity we find in so many, particularly hematological malignancies. They're also not capable of mounting the appropriate responses, and therefore they could be considered as an immunosuppressed group, much as then you would look at organ transplant recipients. I don't quite like that as an answer, because we all know from COVID studies that what we're seeing is the very severe cases of COVID are actually due to an overreaction of the inflammatory response. And therefore, I do think there is something in the observation. What I don't know is, because I haven't seen enough of the data as to what actually is going on in these patients. Are they experiencing this exactly the same massive COVID overreaction that you begin to see in the very severe cases? I suspect you do, and if that's the case, then there certainly is something that'll be very worth exploring in this setting. What is intriguing is this early diagnosis that you're actually seeing. It's in the first year of the malignancy that you're seeing its maximum impact, and it can be controlled for the drugs that are receiving, and this appears to be a phenomenon that occurs out with those patients on drugs. And you can see the numbers on this and the impact of particular therapies also on the Reboot website, which is collecting all the data from different trials to eventually allow proper meta-analytical approaches. You have to be careful with that data because it is biased, because it is for particular patients groups and it's aggregated, and therefore it could be skewed. It's not a population-based study, but nevertheless, for this kind of work, it could give us some very important hints. So I quite like the idea that there could be some overreaction in a more uncontrolled hematological and inflammatory environment.
Thank you for that answer. And perhaps, again, going back to, we have a follow-up question from Agata on the intratumor bacteria issue. Yeah. Are the bacteria found in tumors organ-specific, or are there some found in the tumors, or are the ones that are found in tumors regardless in which organ the bacteria has originated in?
From my reading, it's both. And at the moment that needs to be dissected out, but you know, there may be people who are better able to answer that question, because again, my reading's been restricted to only some of the publications. I have not done a full review of the literature on this topic, but I've certainly encountered both observations.
And during your talk, you also mentioned the issue of e-cigarettes and the potential impacts of those. So would you like to, would you be able and willing to elaborate on your views on this topic? Yeah,
e-cigarettes are a complex issue. Firstly, you have to understand that there's a basis in a big New England Journal study, I think it was in 2016, that reported that to quit smoking, one of the most effective means of quitting smoking was to use e-cigarettes to actually control the craving that exists, because there's both the physicality and the capacity. And that study was absolutely clear-cut. There was a direct benefit. Now, for a smoker, whatever the risks of an e-cigarette are going to be much less than the risk of continuing to smoke tobacco through cigarettes. So that first study shows a real benefit. We now have two real problems. How much do e-cigarettes actually do damage themselves? And the answer is we don't know, because we haven't had 30 years of follow-up to be able to see what long-term consequences are. So we don't know what long-term consequences there are. That's absolutely clear-cut. Second question is, does it encourage young people to be able to take up smoking? We're so busy fighting, particularly in that late adolescent, early adult population, to get them to stop, to not take up smoking in the first instance. So for instance, if you make the accoutrements of e-cigarettes much sexier, much nicer, that they look to be very attractive, both as personalized jewelry or in some way to make it attractive, then it begins to look at as if something, there could be a real problem, that it could be encouraging people to start on e-cigarettes when we don't know the longer term consequence of e-cigarettes. And does it drive them then to cigarettes? The answers to those questions we do not know. For me, there's a third and very worrying development, and that is we're increasingly seeing companies like Philip Morris and others investing heavily in the e-cigarette market. Now, I'm sorry, I just don't trust those industries at all. Their track record is such that they will almost peddle anything in order to increase addiction to nicotine in this way. And once you start putting products like that into the area, then in essence, you're creating a different type of addiction in the population. So all of those three really need to be resolved before you can take a position. Our position at Cancer Research UK has been with the original study that if you are a smoker and e-cigarettes were to help you, then we'd rather you used e-cigarettes and stopped smoking than not to use e-cigarettes at all. That's been the area. But we are very concerned about these other potential harmful effects of e-cigarettes, and hence why it's there as quite a challenge to the community, maybe a different type of community to the molecular community that we might all represent, but a challenge nevertheless, because this issue has to be resolved. And what curbs and controls and policy then needs to be enacted to keep this under appropriate regulatory framework really does matter. I don't know which way that's going to come out. My biggest concern is what are the long-term consequences? You're seeing all these vape shops and things elsewhere. One just goes back historically when doctors used to prescribe a packet of Park Drive or something or Woodbines in order to help clear the chest. It was good for the cough. It wasn't so long ago. So, we get things wrong, and we've got to be careful we don't get this wrong and have a young generation that is adversely affected by something that was being done with the best of intentions. So it's a real challenge to resolve those issues, but it'll be a very different community who will be addressing that, I should think.
So it's a matter of striking the right balance between making it an attractive tool to stop smoking whilst not making it too attractive.
Well, that's what it looks like at the moment. But of course, if we begin to start picking up evidence of harm and detriment, then actually it may not be the best way. And we go back to chewing gum and patches and other support for people who are trying to give up smoking. The problem here is that what's happened, particularly in the United Kingdom, that I find very depressing, is that as cutbacks were introduced even prior to the COVID outbreak, what was the first thing that was being curtailed by local authorities under extreme financial pressures? And I know they have so many where they've got to look after child welfare and schools and everything else, but they started to cut back on quit clinics. Whereas in reality, we know that quit clinics, e-cigarettes, all of these things really help people give up smoking. And probably many of you are not smokers. I did smoke at one point. I know how difficult it is and how those cravings last for so long, and the need for continued support in people. I really feel for people who give up this habit once you get hooked on it. It's not easy. And it's therefore something that the people do need all the support we can get. Tragically, of course, people also have to establish priorities within local government as to how these clinics are supported. So again, this will have a big impact on that whole e-cigarette story. It can't be seen in isolation from that whole cessation program. The good news is some countries are taking a very radical set of steps. Hong Kong in particular, before the turmoil there, was planning on a situation where it was going to make cigarette smoking illegal, period, in the country. And how they were going to regulate that, I don't know. But they'd reached below 9%, and it was mostly in the male population because it was not an acceptable habit in the female population in Hong Kong. So globally, a lot of work going on in stopping people smoking. But from my own experience of working in Africa, I mean, how do you stop the Marlboro man appearing, giving out free cigarette packets and T-shirts in villages in Africa is something I find really quite despicable behavior by these companies. They know precisely how addictive the material they're peddling is, and yet those who can least afford the consequences of cigarette smoke are the ones being enticed into it by this sort of behavior. So, sorry, on World Cancer Day, that's one of the things I do wish we could stop, but I'm afraid I doubt it.
Hopefully there'll be policy-based solutions to that, and there'll be a strong community. Well, there already exists, I'm sure, a strong community.
Oh, there is a very strong community. There's a whole alliance globally in dealing with that. But, you know, I will say it, when you consider that the largest manufacturer of cigarettes is the Chinese tobacco company, which is a state-owned company. At the same time, the same state is actually trying to stop people stopping smoking. It's a crazy situation that you have. And Chinese tobacco now manufactures more cigarettes than any other manufacturer around the world. So, you know, you've got these conundrums of how do you actually tackle an industry which is built around selling a product which is of no benefit whatsoever to the consumer, and actually does real harm, and yet does not seem to wish to fess up to the problems that it has caused.
Thank you. And would you be willing to answer a few more questions, perhaps?
Yes, good. Yes,
so
very happy to.
Thank you to everyone who asked questions so far on the YouTube live stream, and do submit in your questions during the final part of this question and answer session, and I'm sure Professor Borysiewicz will be very happy to answer them. One more personal question. Is there a particular reason that I might have missed why there was a focus on the overlap between dormant cancer cells and the signals used for hematopoietic stem cells and their particular niches? Why do they look at hematopoietic stem cells in particular, as opposed to other kinds of stem cells?
Well, I think the studies that I've come across in this area are that when we begin to look at sites where metastases occur late in disease, or when recurrence occurs late, bone is one of the same areas, and of course those metastases start in bone marrow, so therefore they're in the milieu of bone. And what was reported in the literature here is, and this is a great, a huge amount of work that's gone into this, is that osteoblasts began to show that they can actually play a role of suppressing the activity of such cells, whereas an osteoclastic environment that begins to occur when a metastasis starts growing and therefore eroding bone appears to enhance their growth. And that's why the bone marrow is a particularly important site, because it's one where we know we can find it. It's accessible to be able to obtain marrow samples, to be able to look for rare dormant cells, and we now have the technology again to be able to detect very low cell numbers in that environment. So that works. Brain is another important one, particularly in some of the hematopoietic malignancy. And again, there you now have to really rely on mouse models at the present time. And there's some fascinating studies about how these cells enter into the circulation in the brain, can lie around for a long time, and there they seem to work as pericytes almost to the vascular endotheliums where you actually find them. They don't necessarily all grow, but that's that sort of first session. So I do think we're going to be seeing that the bone marrow is actually, because it's an accessible site and really is important. You can't do repeat liver biopsies or repeat lung biopsies, but you can get at bone marrow. So it's an important site to study. The experimental models will, I think, gradually improve, but you have to remember persistence. Most of the experimental mouse models are pretty aggressive tumor cell types, and you've got to go through quite a few contortions to end up with any sense of dormancy of cells and so keep it suppressed with drugs. So you've got to explore a lot of those phenomena. And I think what we will do is we'll learn a lot. I just would pick those studies because I think we can progress those much, much faster than some of the other sites that we're seeing. So for example, dormant prostate cancer cells, when prostate cancer becomes evident, how often that happens when you begin to pick up bony mets in those individual patients. So we've got hints, and by trying to draw some of those parallels, we can begin to see whether at least the same factors are involved. Now, cell senescence is a really important phenomenon, and the question is does that dormant cell begin to look like a senescent cell? If it does, senescent cells are cleared very effectively by the immune system, by particularly the non-adaptive immune system, the innate immunity that exists, macrophages, NK cells, and others. Because the loss of the conventional markers actually makes them targets for this area. Is it that there are niches that this system cannot actually get at them? They still have to survive. So what is the nature of that tumor senescence? Can we drive cells that are in dormancy towards senescence, and therefore that they can't grow out of that senescence state? But maybe here we're going to get some hints as to how some stem cells get into senescence, and to be able to examine that. And that's the sort of challenge that we set as a grand challenge. That is not, as you know, those are not going to be an easy set of studies to do. We believe to do that, you've got to put sufficient resources into it and a large enough group with different aspects to be able to address the ramifications of that answer. If you just look at any one of those maps that are put up on those slides, you will see just how many different pathways that are going to be involved. The metabolic pathways are involved in the signaling alone of stem cells, and dormant cells are going to be signals. We're touching the surface of some of those changes, changes that are taking place in the genome. And of course, the variation that could occur dependent on the underlying mutations present in the cell depends also at what stage do we get to that level of dormancy. I happen to believe we probably shed tumor cells and dormant cells as something that's an early phenomenon and happen to be there for a long time. But that's more a statement of belief than actually being able to cite the definitive literature which would promote that idea.
Certainly, just thinking of that slide where you showed the different factors that could, in that particular model, drive cells towards senescence. Finding those factors will certainly be a challenge.
I mean, I would particularly direct people to two excellent reviews, one in Nature Cancer last year, and one in Cell. One deals with persistence and one deals with the issue of dormancy. And then if you read those side by side, you begin to see the overlaps that occur, and how then to begin to explore the hematological literature in this area is really intriguing. I think that's one of the most exciting of the challenges. I might even say it's probably one of the most difficult, alongside just understanding what happens to these cells that carry these mutations in a normal tissue. That paper from Cambridge is extraordinary in just showing how many of these mutations are carried by all of us day in, day out, and how rare the actual event of progression to malignancy really is. What happens to these cells? I mean, it's fascinating, to just begin to address that. Oh boy, do I wish I was 30 years younger.
Of course, there's that attitude of, I think that relatively prevalent attitude of oncogenes as being certain mutations that do cause cancer definitively, but that research, that paper certainly shows that it's not necessarily the case, and it's rather the interplay of such factors. So I think maybe just one final question, and just one more thing. Would you mind sending links to those reviews along via email, and then I can pass them on, post them as a comment maybe on the video?
I'm quite happy to send you a list of references if people want to refer to them. And also, you've got my email, so I'm very happy if people want to follow up with anything, please give me a call. I'm not sure I'll be able to answer them, but if I can't, I can at least direct them to people who may be able to.
That's perfect. So I'll just post that as a comment together with your email after we wrap up with the video. So if maybe you could just wrap up with one more question. You also mentioned that one of the main challenges is solid and or developmental tumours in children, and that's a very different type of challenge. So would you mind elaborating on that topic?
Well, what we're seeing is hematological malignancies in children have really made dramatic progress, as we know. That's not without its problems. The treatment of children, particularly with brain irradiation and elsewhere, and long-term consequences really begin to matter. And we've got to remember that as people live longer with cancer, and you've seen that age group that's coming up, we're going to have more and more of the consequences of treatment actually playing out in these populations. But in children, we also have particular types of tumours we see in neuroblastoma that are developmental tumours, and therefore they behave entirely differently. The Wilms tumours, the kidney, they behave differently to the adult type tumours. And there are issues about those tumours that we don't understand maybe as well as we should, because most of us would feel that there's an inherent belief that you should be able to involve signals of maturation and driving to senescence and elsewhere in those tumours in a way that you may not be able to in some of the adult tumours. So understanding their progression, understanding where they come from, and understanding the control factors and how they're going to be interacting, coming from cells that are more to do with development, like neuroblastoma cells, than necessarily cells that are already mature cells come from mature cells in gliomas and elsewhere, would give us new directions in treatment, and potentially learning from that to treatment in adults. So I think it's a real challenge, because we just feel that this area has been underexplored, and there is the capacity for really novel discoveries in this area that are going to have far broader ramifications. I also think they may have real ramifications to our understanding of stem cell homeostasis, which I think is going to be very important in the future. And some of these kids survive a very long time, some don't. So we don't understand the reasons for this. So again, it comes back to that dormancy, quiescence, persistence issue in some of these unfortunate children. We just think it's time that they were looked at in a more systematic way than has been done to date. So I hope it'll at least challenge the pediatric oncology community and those who are investigating that there is a real opportunity that we're committed to seeing if the right studies can actually be pulled together to address this.
And of course, as you said previously, with insights from research in tumours and the effects on T cells and how that could lead into insights into our knowledge of immunology and T cell biology. Of course, there could be various translation aspects into stem cell biology, as you mentioned.
Absolutely. So I think the more we learn, it's always the same, isn't it? The more we learn about any process or procedure, we just begin to understand how this wonderful system that we've got of physiology is a completely redundant system based on redundancy, and how every component, both intracellularly and extracellularly, interfaces with so many different pathways. That built-in redundancy almost makes us far more plastic and able to accept all the challenges, environmental challenges that we face. So there's a brilliance about it in some ways, but disentangling it is a huge challenge. And most importantly, to understand what the primary drivers are that can be converted towards therapeutic benefit, or even markers that can be helping us to achieve an earlier diagnosis of poor prognosis in particular types of use, so we can begin to focus our efforts on what goes on. It's a very interesting clinical observation that the evidence, and Shapiro raises this in his paper, the evidence that just screening for metastasis in clinics by endless follow-up without understanding the natural history of particular types of disease is not really that helpful, because tackling it once you've got radiological metastases doesn't work. But evidence from our own Tracer X studies that Charlie Swanton is leading is suggesting that if we can begin to pick up signatures in the blood, we might be able to start addressing whether we've got resistant clones coming through, and maybe starting treatment long before patients begin to show radiological evidence. Because at the end of the day, if I had to leave a parting message, is remember, it's not the primary that very often kills. It's the metastatic and disseminated disease that ultimately gets to most cancer patients. And that is extremely difficult to treat at the moment. And we need new insights and developments that if we're going to hit this opportunity of getting more people to live with their cancers in an appropriate way.
And it's certainly, those challenges are certainly ones that I hope some of our members and other people tuning in to listen to this talk will take up in the future, or are even currently taking up. So I'd just like to thank you again, Professor Borysiewicz, for that wonderful insight into both translational medicine, the basic science behind various types of cancers, and the current state of basic research, including very recent work that's still ongoing, as well as its potential long-term impacts, as well as including several social issues, including ones arising from the current pandemic. So once again, I'd like to thank you for agreeing to deliver this talk, for answering various questions so comprehensively, as well as to everyone who joined us today and asked questions and took part in this talk in that way. So on Thursday, we are going to have another talk from Professor Stone, who is going to talk about epidemics and mathematical modeling, as well as a talk on the 10th of February from Professor Oliver. And relating to the topic of obesity and potential links with cancer, we are going to have a talk from Dr. Giles Yeo on 10th of March. So I'm sure that I will see many of you there. And I'd like to thank you all again for joining us for another BioSoc talk.
Thank you very much indeed.
I've just ended the live stream. Thank you so much for